Let's have some fun with enzymes. So we're again going to do an experiment, and we've got a hypothesis that says it's possible to measure the activity of enzymes under controlled conditions. And I'm going to introduce you to this enzyme, and we're going to talk about how to do this experiment again. Again, this time we already have an hypothesis, so we're just going to worry about the experimental setup more than anything else. So you've seen this before. In an experiment, we're going to have the different groups, the control groups and the experimental. And we're going to talk about the variables, the independent, the dependent, and the constant. Now, in this particular case, the reaction we're going to look at is the Browning reaction of polyphenol oxidase, where you have a substrate that is converted by an enzyme into a brown product. And if you haven't looked at the lecture part yet, you should go back and look at the lecture part, because things are going to make way more sense if you've looked at the lecture part first. So in order to get this experiment going, every time we need a substrate and we need an enzyme, and then the product is going to be produced. This particular enzyme we're isolating from a potato, because this is where we find this enzyme. And so the first thing I want to introduce you to is this setup. So if we were in class, you would see all these things. And when I saw this picture that Larry put in the handout, I actually started laughing, because there's something very important missing here, which is the top part of the blender, the thing that you're actually going to put the potato in. And I started laughing because one year at like 8 o'clock in the morning on probably a Friday or something, I was chasing all over the lab trying to find that stupid blender, because the blender wasn't on the desk. Where the blender was in the refrigerator, because this enzyme is active as soon as you create it. And so the blender, everything is cold. Everything is kept cold, including the blender, to minimize the activity of the enzyme at times when the enzyme shouldn't be active. So the potato is the star of the show. You chop up the potato, you measure some water in a graduated cylinder, then you blend that whole thing in a blender, and this stuff looks gross. It's this gross looking milkshake. Everybody just goes, ugh. Then you take it and filter it through a cheesecloth with a vacuum filter. And then you have down here a concentrated liquid that has the enzyme concentrate in it. So this is where your activity is. Note that this is already a little brown, and this is going to become important in just a minute. So because there's potato enzyme and catechols in the potato itself, this stuff starts reacting immediately, which is why we keep it on ice during the entire reaction, because keeping enzymes on ice, keeping most reactions on ice, slows down the reaction. This is why you refrigerate your food. Things slow down when they're cold. So the experiment we're going to do is the first thing, and depending on how you want to see this, you can see this as establishing controls or establishing the preferred substrate. So we're going to look at the control groups here first, and then there's actually a couple of experimental groups in there. When we do this in the lab, I have to be very clear to my students that we add things in order, and it's very important to add the enzyme last, because the enzyme is going to start working the moment it starts working, and the moment it hits the substrate. And since there's an accumulation of brown that keeps on going and the enzyme does not get used up, there's actually a time dependence on this whole thing. So we got to be very careful about setting this exactly right. So you add 7 mils of water, then you add 5 drops of water here, additional water, and then in A you drop 5 drops of catechol, In C, it's 5 drops of catechol. In D, 5 drops of phenol and 5 drops of hydroquinone. And then you add enzymes in 4 out of these 5 things. Catechol, phenol, and hydroquinone look very similar, right? They all have this ring structure, this phenol ring structure, and there's a couple of oxygens attached to it. So one of the things we want to figure out here is how specific this enzyme is. Can the enzyme tell a difference between those 3 structures that you and I probably can't really tell a difference? So the first thing we're going to have to think about is, what are we looking at here? Which ones are our controls and which ones are our experimentals here? And I'm going to show you. So think about, well, actually, let's think about this for a moment right now. In order for this experiment to work, we need a substrate and an enzyme, right? So something that doesn't have a substrate, that is going to be one of your negative controls. Something that doesn't have an enzyme is also one of your negative controls. Now, we know that the substrate here is catechol, so catechol is your positive control. And phenol and hydroquinone, those are your experimentals in this case. So after you incubate this substrate and you do this incubation for 5 minutes, this is what your results are. This is what you see. And you quickly establish that your positive control is brown. Your no substrate negative control is pretty clear. Your no enzyme negative control is also pretty clear. And neither the phenol nor the hydroquinone have, which are both experimentals, have much of any kind of browning that's happening here, which suggests that they are not good substrates for this enzyme, which in turn suggests that the substrate is very, very specific. Now, being able to tell the difference between those tubes is quite hard. And one of the things that makes this especially difficult is that there are other substances in there that create different colors. So, for example, this thing here, B, that doesn't have any substrate, should really not have any kind of color at all, but it looks like it's a little darker than even the C, which doesn't have any enzyme, right? They should really not be any different. Well, that's because the enzyme itself, the potato part, there are other things that are happening in there, and that actually causes a color change that's not brown. It's just a little different, but our eye can't distinguish that. So the next thing I want to introduce to you is the spectrophotometer. The spectrophotometer gives you the ability to see things that you normally wouldn't see. You can specifically say, I want to see only something at 540 wavelength, which is what this brown color is, and we're going to measure this only at 540. Now, when we do this, the next thing we're going to have to think about is which one here, which one of these, because you always have to, in a spectrophotometer, you always compare things to one another. So you have to use your first thing as a blank. And the question is, I'm going to go back real quick, what's the best thing to use as a blank? You would use as a blank one of the negative controls. So you could either use the no substrate control or you could use the no enzyme control. Since our enzyme, as we see it in the little tube, since our enzyme is actually a little bit brown already and there's some activity there, the best control is C, because it doesn't have any enzyme in it and water is clear and substrate is clear. Oh, that brings me back to do you know why we added the 5 drops of water here? We added it because we wanted to make sure, remember where you want to keep everything constant except for the one thing you're changing? So the only thing we wanted to change here was the substrate. And since it matters, concentration-wise, how dark something is, we needed to make sure that the volume in each of these tubes is identical. So in the places where we didn't add substrate, we added a little extra water. Same thing with the enzyme, we added a little extra water. Okay, so back to here. So we used C as our control, and we used that as a defined 0. And I actually put that number wrong. That's minus. That is a negative number. I messed that one up, so that I'm going to have to correct that. Okay, so then you put the blank in there, and you close the lid, and then you measure. And what you measure is absorbance. The spectrophotometer shines a light through your tube, and it tells you how much light is being doesn't come through, how much light is being stopped. And so these are a little bit out of order, so you got to be a little careful here. So C is your 0, so C comes in as 0. S1, that's B, that looks pretty clear, but it looks actually darker than the C, but it actually is a negative number, so it actually absorbs less than C does. S2 is E, that's almost no absorbance whatsoever. S3 is D, there's also almost no absorbance whatsoever. And A is the only one where you have absorbance. So the spectrophotometer allows you to distinguish between colors that are very similar for your eye. And it allows you to see differences that you would actually probably get this wrong. You would say that the B is darker than the C, but it really isn't. All right, the next thing we're going to, we're first here, we're going to have to think about the dependent and the independent variable. So in this particular test, we did an enzyme, the polyphenol oxidase test, and we tested for the presence of catechol or the presence of a substrate, right? Your positive was a dark brown color. Your negative was a clear color. You're measuring the color, and you're measuring that with a spectrophotometer. So you can either measure color or you can measure absorbance. The variable you held constant, except in the control where it wasn't in there, was the enzyme. You also held constant the tube, the volume, the procedure, the temperature, the pH, the length of time. And the only thing you changed here was you changed the substrate. You tried for different substrates to see which substrate was the best one. And it turned out catechol was, in fact, the best substrate. So you can use the same experiment and change different variables. So we're going to actually use the same experiment, the polyphenol oxidase, to ask different questions. Because now we're going to ask questions about the enzyme itself. So the next hypothesis we're going to have is that the enzyme extract will convert more catechol to a measurable amount of benzoquinone when incubated at 40 degrees Celsius or 60 degrees Celsius, and significantly less at temperature extremes. Before we go much further, I want you to make sure that we understand what 40 degrees Celsius is and what 60 degrees Celsius is, because most of you are probably not very comfortable with temperatures. So 40 degree is about your body temperature. 37 degrees Celsius is your body temperature. So 40 degrees, that's about body temperature. And 60 degrees is warm enough that you can touch your wrist with it without burning yourself. So think about here, what is your dependent variable, what's your independent variable, and what variable are you holding constant here? So we'll go to the next slide. The way the setup works here is we're modifying the temperatures. We're keeping the catechol the same, So we're using the same substrate each time. We're using the same amount of enzyme. We're actually letting the water bath reach temperature, to make sure that they're all at a constant temperature. And the things that we're varying in this experiment is the temperature of the water bath. So here you see, after all these things are added to each other, this is where they are in the water bath. So there's an ice water bath, there's a room temperature water bath, there's a 40-degree water bath, a 60-degree water bath, and then there's a boiling a thing where everything is boiling. And we do this for 5 minutes. And at the end of the 5 minutes, we look at the results. And at 0 degrees, we have a little bit of a temperature. We have some browning. So looking at the numbers, the one that's the brownest over here is S4, which was the 60 degrees. S3, which is the 40 degrees, was second. S2, which is 22 degrees, that's room temperature, was third. And then S1, the 0 degree, also worked some. And the one that didn't really work at all was the 100 degrees Celsius. Now, the 100 degrees Celsius, that's boiling. And if you think back, when I was telling you earlier about the activity of enzymes and denaturation, 100 degrees is too much for an enzyme to handle. Just like you can't boil an egg without denaturing the egg white into the solid, you can't. The enzyme will not survive this. Now, we want to test for that. So both of these samples, the 0 degree and the 100 degree, they both didn't have much of a reaction. And so what we did after this is we took these two samples and we incubated them at 40 degrees for another 5 minutes to see if the enzyme only slowed down, because at 0 degree you didn't have much of a reaction, but you may still have enzyme there that you could still recover. And at the 100 degree you didn't have much of a reaction. Again, the question is, do you still have enzyme there, or is the enzyme just broken? And so after 5 minutes, we looked again, and it turns out that the enzyme that was incubated at 0 degree recovered perfectly fine. As a matter of fact, it did as well as the 40 degree sample there, whereas the sample that had been at 100 degrees did not recover. Because the enzyme is broken, it's denatured. Now, if we think about what were the positive, negative control, and what were the independent and dependent variables here, your positive and negative controls. Your positive control was the 40 degree. There wasn't really a negative control because we'd established that earlier, and so since we're doing this with the same substrate and the same enzymes and the same everything, from now on we can use the positive control and the negative control that we actually established earlier. So in terms of dependent, independent, and constant variables, this time around the enzyme stayed constant and the substrate also stayed constant. We again measured the color, but this time the variables we changed was the temperature. Now we're using the same experiment by modifying different variables. We're utilizing it to measure different things. So this time we're looking at enzyme activity at different temperatures. The next experiment is we're going to look at enzyme activity at different pHs. And again, we're going to say, the hypothesis is that these enzymes are going to be more active as a pH of 7 or so than they are at other pHs. So we're going to set up the same thing as before, except this time we're not going to use water. Instead of the water, we're using pH solution. So we have a pH, we have a solution that has a pH of 4 and add the catechol and the enzyme, one that has a pH of 7, add the catechol at the enzyme, and one that has a pH of 10 and add the catechol at the enzyme. And this time when we're looking at the absorbance, you'll see that the one that does the best is the one at pH 10. So this enzyme is pH dependent. Because the pH, because the pluses and minuses, the positive charges and the negative charges, they change a little bit. The active site isn't quite as good and quite as active as it would normally be at a changed pH. It still works, but it just doesn't work as well. And when we're going to look at what is your dependent, your independent variable here, in this case, we changed the pH. So the independent variable is your pH. This time we held the enzyme, the substrate, the temperature, and the tube, the procedure, the time, all these other things constant, and we only changed the pH. So this time we were testing for enzyme activity at pH. And you're measuring the same thing. You were measuring color all along, or absorbance as a stand-in for color. Now the next thing I want you to think about, and I may grab a different slide for this. Hang on, give me just a second here. If I can get to that. Stop sharing this real quick and share a different, hang on, let me see. Because this one is best explained in a different way. Share, hang on, I'm gonna open this up. Where is it? Second, this one. All right, this time I'm gonna share with you an application so I can have activity going on in my, I'm gonna share this with you. Come on, let me do it. Okay, hide this. And go back here. Okay, so when you look at an enzyme, I like to think of enzymes as turnstiles. It's just things that are churning things over. And when you look at the substrate concentration, and you're using substrate concentration, is going to be the variable that you change. So that's going to be your independent variable. And your dependent variable is going to be on the y-axis. So substrate concentration is going to be on the x-axis. The rate of the reaction will be on the y-axis. If you have just a little bit of substrate and you have more enzymes and you have substrate, your reaction is just going to be constant. And then if you increase the substrate, the reaction will increase until you get to a point of saturation where you have all the enzyme sites, all the active enzyme sites being utilized. And then it doesn't matter how much more substrate you add to it, you're not going to be able to make it go any faster. That's exactly the same way as when you're trying to get 100,000 people into a stadium and you only have 2 turnstiles that can let 1 person through a minute. You're not going to get people in any faster because the turnstile is your rate limiting step. Here you have the opposite basically. So you're not, this time what you're changing is the enzyme concentration, not the substrate. And again, you got to be looking at what's your rate limiting step. So if you have a bunch of substrate, and you only have one turnstile that will bring people in. It doesn't matter how much substrate you add. The only thing that's going to change anything is if you add enzyme concentration. And if you add enzyme concentration, the more enzyme you add, the more your rate of reaction is going to go up. So if you increase your enzyme concentration on the x-axis, your rate of reaction will be increasing on the y-axis, which means your rate of reaction will keep on going up and up and up, provided that there's sufficient substrate. If you don't have sufficient substrate, then at some point you're going to reach a plateau. And that's pretty much what I wanted to show you. I have this in here. Yes, I do. So for your homework assignment, you have all of these activities in all of this is explained in this handout, and there are questions in between here that I want you to answer. And so answer these questions and then submit them at the activities. And that's it for this part of this week's class.